This video will explore the diverse metabolic pathways found in microbes, which are essential for their survival and growth. Metabolism includes anabolism, the process of synthesizing essential biomolecules, and catabolism, the breakdown of nutrients to release energy. Anabolism involves the synthesis of crucial biomolecules, such as carbohydrates, lipids, amino acids, proteins, and nucleic acids, which are necessary for growth. Catabolism involves breaking down nutrients and stored compounds to release energy. Key catabolic processes include glycolysis, the tricarboxylic acid cycle, and fermentation. Adenosine triphosphate, or ATP, is the chief energy carrier molecule. It is required for anabolic processes and is formed during catabolic processes. The trapping of chemical energy in the form of ATP is called phosphorylation. There are three types: photophosphorylation, oxidative phosphorylation, and substrate-level phosphorylation. Photophosphorylation occurs in the presence of light in photosynthetic organisms, trapping solar energy in ATP. It can be cyclic or non-cyclic. In cyclic photophosphorylation, electrons return to the same chlorophyll molecule, forming ATP. Non-cyclic photophosphorylation involves electrons being received by NADP+, leading to ATP formation and oxygen release. Oxidative phosphorylation involves passing electrons through an electron transport chain to generate ATP. It occurs in the inner membrane of mitochondria in eukaryotes and the plasma membrane of prokaryotes. Substrate-level phosphorylation generates ATP by directly transferring a high-energy phosphate bond from a metabolic compound to ADP. NADP+ and NAD+ are essential electron carriers, serving as the cell's reducing power. They act as coenzymes in many oxidoreductase enzymes. Glycolysis is the initial phase of glucose metabolism, partially oxidizing sugar to pyruvate. The three main routes are glycolysis, or EMP, pentose phosphate pathway, and Entner-Dauteroff pathway. Glycolysis, or anaerobic fermentation, is crucial for obtaining energy in the absence of oxygen and is present in both aerobic and anaerobic organisms. Some bacteria, like E. coli, use the EMP pathway for aerobic carbohydrate catabolism. The non-phosphorylated ED pathway is seen in archaea. The overall balance of glycolysis shows that glucose yields pyruvate, ATP, and NADH. Pyruvate is further converted depending on the organism's conditions. Glycolysis involves 10 reactions catalyzed by 10 enzymes in the cytoplasm. It is divided into two stages, the first utilizing ATP and the second synthesizing a net of 2 ATP molecules. Polysaccharides, like glycogen, can enter the glycolytic pathway by being converted into glucose 6-phosphate. Monosaccharides, such as fructose, can also enter glycolysis by changing to glyceraldehyde 3-phosphate. The methylglyoxal pathway is an alternative to the EMP pathway. It operates under low phosphate concentrations in bacteria and results in no ATP generation. The Entner-Dauteroff pathway is an alternative for glucose oxidation to pyruvic acid, found in some gram-negative bacteria like Pseudomonas and Agrobacterium. The ED pathway occurs in prokaryotes and uses specific enzymes to create pyruvate from glucose. The Entner-Dauteroff pathway yields a net of 1 ATP, 1 NADH, and 1 NADPH per glucose molecule, which is lower than the yield of classic glycolysis. In the ED pathway, glucose 6-phosphate is converted to KDPG, then cleaved to glyceraldehyde 3-phosphate and pyruvate. This process only generates 1 ATP molecule. The partial non-phosphorylated ED pathway is found in certain bacteria and archaea, where an intermediate product formed before KDPG is non-phosphorylated.